from Maryland, and we had heard she had trained a dog to smell the hives for American foulbrood, and so we thought that was great. We'd save ourselves a lot of lifting just to have the dog, and we were trying to find out from her what she was doing. But since that time, she went to Penn State, got her degree in agriculture, and then went on in agricultural entomology and got a master's degree at Penn State. She's out there with the extension with Penn State, and she's one of the world's leading authorities on CCD, which I know everybody is out there right now, and this is the hot topic. We're very fortunate to have gotten her because I'm sure she could go almost anywhere in the country, and people want to hear about CCD. So we are very happy to have her here today to speak to us on two different talks with a break in between. On the first one on chronic collapse disorder, and then on bee nutrition, which ties right in. So at this time, it gives me pleasure to present our speaker, Maryann Frazier from Penn State.
Well, can you all hear me okay? It's not working? Do I need to turn this on?
No, you're all set. I'm just turning you up slowly.
You're slow and turning me up slowly. Okay, hopefully eventually you'll hear me all right. I first have to say how gratifying it is. As a speaker, we move around, I move around the Mid-Atlantic region a lot, and I talk to, small groups, large groups, but it's always a lot more exciting to speak to a large, enthusiastic group. And I'm really impressed with this group and all of the things that you've been able to accomplish, and the energy and enthusiasm. And I have to thank Ken for, first of all, that great introduction, and for asking me to come to the meeting, inviting me, and also to Bob for picking me up at the airport yesterday. My flight was a little bit late, and he was very patient. And Mary and group who took me out to dinner last night. It was lovely, lovely meal. I really enjoy it as a speaker. It's so nice to go to a place where you're welcomed and taken care of, and you have a great enthusiastic crowd to speak to. So I really appreciate that. So somehow I moved right along here. Let's go for it. So yes, I am one of the team that's working on the colony collapse disorder with a lot of other people, and it's been really exciting as a scientist and extension person to work with such a wonderful group of people and to collaborate on such a very significant problem. So let me just get right into this for time's sake, because I want to give you some time to ask questions at the end. So colony collapse disorder, I bet there's no one in this room who hasn't heard of colony collapse disorder. Is that correct? Has anybody not heard of colony? That whole issue, the phenomena of people knowing, not just beekeepers, but non-beekeepers, being interested and knowing about this situation and being concerned about it, has been an interesting ramification. We have been totally shocked by the media's and public response to the decline of honeybees. And in some ways, it has been a very unfortunate situation, but on the other hand, it has served to make people, non-beekeepers, so aware of bees and their importance in the food system. So some of you may know the story of sort of CCD, and Dave Hackenberg, who is a beekeeper in Pennsylvania but migrates to Florida, was the beekeeper who originally notified us in November of 2006 that something was wrong with his bees. He had seen a huge collapse in splits that he had made in Florida. And Dave's been keeping bees for 50 years, and he is not one to, cry wolf. He recognized, even though beekeepers over the years have been having increased losses due to mites and resistance to miticides, etc., this situation was something he had never seen before. So he immediately contacted us, brought some hives up from Florida, some nukes, and we just kind of got the ball rolling on the whole issue of this decline. It wasn't even called CCD at the time. Is that okay? Well, no.
No.
Oh. it's a little hard to see.
Yeah, you're
I'm
dark and that's really bright, so the whole thing is you want to keep this bright, but you also need some kind of light on the speaker or you just come off.
Well, you don't need to see me. As long as you can see the slides, you're fine. Yeah. I want people to be able to see the slides if that's okay. Yes. Don't worry about me.
How about a side one over here? Right side.
That's fine. That's good. That's fine.
Is that okay?
There. Huh?
That's good.
Is that all right? No? I'm going to...
That's good. That's good.
That's great.
I'll try to stand aside here. Okay. I just want to sort of quickly, a lot of you may know this already, but I want to just sort of review what makes CCD unique. I mean, we've had a lot of collapses of colonies in the past before. We've had bees die in large numbers across the country for a number of reasons, including, as I just mentioned, tracheal mites, varroa mites. Then, of course, we had resistance to the materials we were using. We saw, large decline in bees for those reasons. But this situation is different in that, first of all, the bees build up very quickly, and the colonies are quite large. They look good. Within a matter of a month, and sometimes within a matter of a week, the adult population in these colonies just totally disappears. And this ratio of adult bees to brood is totally out of whack. This is a colony that you can see here. The number of bees here could never support the number of adult bees, that brood, could never have produced that brood. So this whole idea, typically in a colony what we see is the adult population is probably about twice the population of brood, capped brood. In this CCD situation, the situation is pretty much, the circumstances are kind of reversed. The brood is 2 or 3 times more than and sometimes there are no adult bees. The adult bees are totally gone. And sometimes there'll be a queen and a very small number of bees that have just obviously recently emerged, very young bees. So the rapidness with which the adult population leaves, the fact that it happens so quickly, so few adult bees, the amount of capped brood. There's also this phenomena that maybe isn't totally consistent, but does happen, where these colonies then are not robbed out by other colonies. They're not invaded by wax moths or small hive beetles, which is very unusual. In these circumstances, in these yards where these guys have hundreds of colonies, and there are some that do remain that are strong, they don't rob the weak colonies. And that's an unusual situation. So last year, in the midst of all this, we were trying to figure out how significant is this, what's going on, is this a real phenomena, is it something that's just happening this year, are the losses really as significant as we think? So Jerry Hayes from Florida, Dennis Van Engelsdorf, Dewey Karen very quickly did a survey, and you may have seen this in the American Bee Journal, to estimate what was going on. And they did this very, very quickly, responded very quickly, and they had a criteria for what constituted CCD and what didn't. And in a nutshell, what they found was that operations that did not have this phenomena, this CCD phenomena, lost about 25% of their hives last year. Those that did, that were designated as CCD operations that had this phenomena, lost 45% of their colonies. Overall, last year, we figured we lost about 1/3 of the hives in the United States. This year, Jeff Pettis has done a very quick and dirty survey of about, so far, he's calling beekeepers, large beekeepers, and getting an assessment of what, okay?
Yeah.
Assessment of losses, and he's figured he's surveyed counted for about 10% of the hives in the United States. And the losses this year are slightly higher, about 36%. The guys in California, the Western beekeepers, are having very big problems with the bees in almonds right now. They're particularly the guys from the West. We've had a number of people from the East go West. Their bees have held up pretty well, my understanding, but the Western beekeepers are having a lot of loss. So there has been a lot of interesting suggestions. It's unbelievable the people who are concerned and have ideas about what is happening with our bees. And of course, one of the biggest, most highly publicized possibility was cell phones. All this cell phone use, tremendous amount of cell phone. Well, this is Charlie Borsak, a beekeeper in Pennsylvania. He did this little experiment. He put these cell phones in front of the entrance to his hive, and he turned them on, made sure they were fully charged, and the bees, it didn't bother the bees at all. We also know that there's a lot of places where there are no cell phone towers, in the Midwest, for instance, and they've had significant collapses. So we know that cell phones are not the problem, not causing CCD. But here's another interesting perspective from one of our Maryland beekeepers. I never let my bees use cell phones. They are social insects, and I have found that once I let them use cell phones, it's impossible to control their use. The charges from time overruns can bankrupt even the most efficient apiary operator. Once they learn to use them, they become dependent. They stop returning to the hive to dance and just phone in the location of their origin. Furthermore, the increased peer-to-peer communication plays havoc with traditional bee values. To heck with pheromones, they say. Chemical communication is passé compared with digital. As in many societies, the young are the early adopters, spending their time text messaging instead of doing their jobs. In the end, we observe a breakdown in hierarchy and a fatal anti-royalist sentiment. This, then, is the cause of CCD, foolish notions of independence among the immature, loss of authority of the elders, and breakdown of group cohesion, and the collapse of the aristocracy. So that's Laura Kaufman. It's really amazing that people have been able to keep their sense of humor in the face of this crisis, essentially. But what we have done is again, very quickly, a lot of conversation with a lot of beekeepers who were having this problem. And a group of individuals came together at a number of the meetings last year, and we tried to very quickly prioritize what do we think really is most likely going on here, and what basket should we put our research eggs in. And pathogens, we know, are a problem in hives, and we've seen an increase in pathogens, particularly with the introduction of mites and their ability to break down the immune system of the bees. So oftentimes new pathogens introduced can also, cause this kind of dramatic, just like if you think about the early American Indians, when the American or the European colonists came, there was a big loss of Native Americans because of the introduction of diseases that they didn't have any defense to. So a new pathogen, or a pathogen that somehow had changed and became more virulent to the bees, was one of our big concerns. The other was pesticides. There hasn't been a lot of looking at pesticides, although we were suspecting that either environmental pesticides, like agricultural use pesticides, or the ones that the beekeepers themselves were applying might be playing a role here. Also environmental stress, things like the genetics of the bees, as well as the lack of nutrition. We're very concerned about the poor nutrition that our bees are receiving. And I'll talk more about that later today. So these were kind of the top tier things. What did happen too is very quickly things were rolled out like high fructose corn syrup because of the survey that was done among these beekeepers. And we found that some of them had used high fructose corn syrup, some of them had used sucrose, and some of them hadn't fed at all. So it kind of didn't make sense that high fructose corn syrup was the cause of this. We also, in the study that I'll talk about, this big CCD study, we looked at varroa mites, tracheal mites, nosema, and none of these things seemed to be consistent. There wasn't, some beekeepers had high levels who had CCD, some didn't have high levels of one or the others of these things. So those kind of things we kind of looked at, but we didn't consider them the top line of investigation. So I want to bring your attention to two publications that have been come out recently. Actually, this one in 2007. This is actually a book, and it was commissioned by the National Academies of Science, which is very well-respected group of scientists. And they looked at this. You can read a synopsis of this online. If you go to the National Academies of Science and look up pollinators, there's a summary of this that you can print out. But there's also a book that you can buy. And what they looked at was the status of all pollinators in North America. And in a nutshell, what they found was that pollinators in general are in decline. This was even before CCD. This was their finding. And it was amazing to hear May Berenbaum, who some of you may know, who's a very well-known entomologist, University of Illinois. I was at one of these early meetings on CCD, and she kept saying, well, we talked about that, and this just is exactly an example of this decline of honeybees, is an example of what they talked about in this book documenting the decline of pollinators overall. Interestingly enough, in Europe, okay, there we go. Boy, this thing's reacting, I'm kind of falling asleep. Okay. In Europe, there was an interesting study done in 2006 where they did a large survey of pollinators, insect pollinators, in the Netherlands and in Great Britain. And what they found was not only were certain groups of pollinators in decline, but the plants that were very dependent on those pollinators for pollination, sometimes they're, with honeybees, we're lucky, they're very generalist pollinators, but there are some bee species and some species of moths and hummingbirds and so forth that are, I don't know about hummingbirds, but certainly moths, that are very tied to a particular plant. And that plant requires that insect to visit it in order to have adequate pollination. So they found that both the insect pollinator and the plant were in decline. So what happened with the CCD? We were very, very fortunate because we came together as a group. We talked about what we thought needed to get done. We got emergency funding from the American Honey Board. They were, the National Honey Board, excuse me, they were extremely critical to this situation, and they were very generous. They don't have a lot of research funding, but they put the money to this for us to be able to quickly go out and sample these colonies, sample non-CCD operations, bees, brood, pollen, wax, assess the health of these colonies, and then bring these samples back and divvy them up. Thank you. Mary, thank you very much. Divvy them up between the team to then analyze these samples and try to figure out what was going on. So these are there were control colonies, and they were identified as strong, weak, and then there were CCD colonies that were CCD operations that were identified as either strong, weak, dead, recovering, packages added, or an installed package. And these were the number of colonies from each of those circumstances. So here's the group actually doing their sampling. And this is a picture out in California where, I'm sorry, I haven't actually had the opportunity to experience this myself, but apparently the numbers of colonies out in California at this time of the year are just incredible. About a third to a half of the bees in the United States actually go to California for almond pollination. And there's a tremendous amount of acreage actually under, they're expanding the almond production. There's new trees being planted. I don't know exactly what the acreage is, but it's really increasing very, very quickly. So the demand for bees, and almonds are 100% dependent on bees for pollination. So there's a tremendous demand for beehives out in California at this time of the year. They're in the orchards right now, the bees are. Which made it actually quite easy for us to sample because the bees were concentrated in last year, in January and February in Florida and in California. So the folks who went out and sampled were able to sample a lot of operations from different places in the country by being just in California and in Florida. So that was really helpful.
Maryann, give us a number on that third of the bees in California. What number do you have?
Well, there's 2.4 million colonies in the United States right now. That's the number that we typically, and about a third to a half are go out to California for almonds.
About 1.25.
About something like that, yeah. So 1.5 million colonies move out, something like that, to California. So, so after these samples, then, as I said, from these different operations came back, some of them went to Jeff Pettis at the USDA lab in Beltsville, and they did screening for varroa mites and nosema, and you've probably seen that there's a real big increase in nosema cerani. That we used to think our nosema was nosema apis, but as they started to look and really be able to genetically identify the nosema, it turns out that the nosema that we have, for the most part, is nosema cerani. And how this happened, we're not exactly sure. Did it slowly turn over? Have we all these years been misidentifying our species of nosema? Nobody's really certain. And there's a lot of concern about nosema being responsible, nosema cerani being responsible for high losses in other places like Europe, Spain, for instance. They blame their high losses on this increasing presence of nosema cerani. But, so that there were samples that went there. There were samples that went to Dave Carpy in North Carolina to look at the genetics of these bees. Was there one particular genetic line that was susceptible and falling apart, which wasn't the case. Again, Jeff did this work with nosema, with varroa, and with tracheal mites, and they didn't find any consistency. The control operations in the strong colonies had levels similar to the declining of the CCD operations. Another group of samples went to Diana Cox Foster, who's an insect pathologist at Penn State. She teamed up with Ian Lipkin at the university, at Columbia University, who is a very powerful, highbrow human pathologist, works on human diseases and emergent diseases. They got very interested. This is one of the ramifications of this lot of publicity that we got. They actually contacted us and were interested in this problem. And they had a lot of expertise and a lot of technology that wasn't available to us as insect pathologists. And so Ian and his group worked with Diana, and they analyzed these samples of bees, adult bees, for diseases. Am I standing in the wrong place?
No,
here?
I think it's battery.
Oh my goodness, I can talk loud. Okay. That's okay. Okay, I'll just speak up. Can you all hear me okay? I'll just talk
I'll get you back up in a
Yeah,
sec.
take your time. I hope I don't blow anybody's eardrums off when that comes on. But what they did was they used these things called gene chips. It's like a little, they did a couple of different things. One thing was they were able to look at the genome of the bees, and they were able to identify it against the genome of the honeybees, since we know that. So anything that was associated with the bees, they could recognize. And anything that was associated with known diseases, they could pick that out genetically. And this is, not my area of expertise, so I hope I'm explaining it okay. Anything that was different, they could see that there was something different, that there was something that was not known, not recognized before. So they were able to identify quickly everything, all of the pathogens that were known, as well as the pathogens that were something that was new, something that hadn't been there before. So what they found was in these CCD operations, non-CCD operations, was this they found a number of things. They found cashmere bee virus, nosema apis, nosema cerani, and then they looked at where the combination of these things was. And in 83% of the CCD operations, they found this IAPV, this Israeli acute paralysis virus, which is something new that hadn't been identified before. It had, not in the United States anyway, it had been identified in Israel, I think in 2004 was the first publication on this, the first find of this. But these bees had high incidence of this IAPV. The other thing that's interesting here is that these bees were also very, very, the CCD operations really had, they had all of the bees had those operations had high levels of a lot of pathogens. There was a lot of infection, in other words. So lots of high levels of different kinds of pathogens in the CCD operations. So what this indicated to the researchers was that these bees are sick and they're very susceptible to the diseases that are in. They're not able to fight them off, in other words. So we saw that particularly in the CCD operation. Again, the two important things were the amount of infection that was in the CCD operation and this new IAPV. The thing about the IAPV is that it was also found in all of the Australian packages. And we're pretty certain that the Australian packages came in with this IAPV. We don't think it's the only way that it came in to this country, but it is most likely one of the routes. Well, as you can imagine, the Australians are not all happy about that situation. And there's been some real controversy. And there has been a real effort by the national groups to try to block, until we figure this out, the importation of Australian packages into the United States. But APHIS has been unwilling to do that, which is kind of interesting.
I'm not sure. What is the name? What is those initials that you just...
Israeli acute paralysis virus. Israeli acute paralysis virus. That's what it's called. And they abbreviated it as IAPV. Sure, question?
How much CCD do they have in Australia?
They don't have it. That's part of the reason why, at least from everything we've heard and the beekeepers that we've talked to and the officials that we've talked to, they do not have this CCD. And that's why we don't think, in part, why we don't think that this virus alone is responsible for CCD. We don't think that virus alone is the cause of CCD. But it is very predictive of CCD. 83% of the colonies had this IAPV. So it doesn't necessarily mean it's the cause, but if you have IAPV, chances are you have CCD. So it's predictive.
Well, you were just saying they were doing the gene mapping.
Mm-hmm.
Now, were they able to determine if there's a genetic predisposition to a lower immune system? Is that what it is? Is that there's a genetic predisposition to have trouble fighting off diseases and there's a new disease?
Yeah, no, I don't think so. I don't know that they've been able to look at the genetics at that level. You know, they've been able to say, is there a line of bees that are susceptible to, and they haven't found that. You know, the genetics are very highly variable. So there's Dave Tarpey has found that there's no one genetic group that seems to be predisposed to CCD, from my understanding of what his work has been.
Maryann, do genetically modified foods fit into this equation?
Yeah, let's talk about that. Let's hold off on that question, because we're going to talk now about the pesticides and about, the question was about genetically modified organisms. So let's hang on to that thought for a minute, and we'll talk about that here at the...
Just one thing I want to ask, which I'm a little confused on. At the beginning, you said with the CCD, the bees are gone, but you said there's an awful lot of brood. I was under the impression that there was nothing left in the hive, no brood or anything.
No. Oftentimes in dwindling colonies, that's what happens. But in CCD specifically, there's a lot of brood left. That was one of the things that made this so different from other dwindling, you know, spring dwindle, fall dwindle, autumn collapse. There's been a lot of different dwindlings in the literature for different reasons. But the difference with this is that typically the colonies are strong, the adults leave, and they leave behind a lot of capped brood, which is very weird.
That is because I figured everything was going the way I understood it. You know?
Yeah, no. In most cases, there's a lot of brood left. And the brood appears to be healthy. So it's the adult population that for some reason leaves the hive. And leaves behind all that, you know, for bees, as we all know, that's a very odd circumstance for adult bees to leave behind a nest of brood like that. So, question?
Oh, yeah, no, we've had to see the percentage of IATV was non-CCD.
That was only one operation did not have, that had CCD, I'm sorry, one non-CCD operation had IAPV.
Had it, okay.
Had it, that's right. That's what this one means. There's the non-CCD. It was one operation, 4.8%, on a percentage basis, but it was one operation.
I have a question.
Uh-huh.
So, I know we often talk about consequences, but a lot of us are still a little bit confused about whether hives really failed for some other reason you're calling it collapse.
Right.
So, I mean, is a clear sort of list of symptoms, has that emerged to the point where you could say this is and this isn't? I mean, does it only happen at the height of the season? And, like, can we not ever blame our failed hives in the middle of winter on pollen?
Well,
Clear yet?
that's a very good question. And the problem is that this whole thing was sort of found in places like Florida and California where the bees were active and rearing brood. The question is, could the same, like in Canada, for instance, they insist they don't have this problem. But the problem in Canada, of course, is that the bees over winter, if they're dying, and if this thing may be going on, whatever this thing is, they may not have had the opportunity to rear brood. Our typical definition of this is that the ratio of brood to adult bees is out of whack, that there's twice as much brood as there are adult bees, where it should be the other way around. But if you're coming out of a winter situation and the bees haven't reared much brood, or in some cases any brood, is it possible that this CCD thing could be happening? It just doesn't fit the symptom. And that is a question that we don't have the answer to. So could some of our bees that are coming out of winter be CCD colonies? It's possible. We don't really, it's only we're into the second year of this, and we're still trying to work out some of the details. It does seem, I will say, this whole thing in general, though, what we have learned is that our bees, there's a lot of disputing CCD, but there's no one disputing the fact that our colonies are in poor health, and that in general, we are seeing a decline in honeybee health and in honeybee populations. The CCD thing, in my mind, is the latest ramification of a very sick species. That bees in general are in poor health, and it's in part because of mice, because of poor nutrition, because of stress, because of pesticides. And then there's something else that happens in addition to all of that most likely ends up, and maybe it's this IAPV that kind of breaks the camel's back and ends up being something that we call CCD. But it is a little foggy yet. It is a little foggy. But we have tried to take the focus in our work off of CCD and put it onto decline in honeybee health. And what can we do, regardless of CCD, to fight this problem with declining honeybee health?
Do you have any more information on the cell phone? I know you said that you don't think it is that, but do you have any more information on the possibility that cell phones can be attributed to this? Because they came out several years ago. And
right,
they came
yeah.
out with a lot of bees.
Well, the thing is, they've been using cell phones even in Europe for longer than we have, and there's much more dense use of cell phones in some places, and in some places they've lost bees, in some places they haven't. We just don't see any correlation between.
This last summer, I spoke to someone who was in a remote area of Massachusetts. They couldn't even get me on the phone because there was no ability on account of the satellite could not reach the phone. When they came out of this remote area, then their phone was fine.
Yeah.
But from what I understand from that person, there were a lot of bees in that area.
That were dying?
No,
that were
not.
Alive. Yeah. Well, and likewise, there's places in the Midwest where there's not much cell phone coverage where we see a lot of this collapse. So that's why we don't think that there's a correlation, at least not that we can see. Not that, again, what we've been able to do is, what we've tried to do is prioritize what do we think is most important, and not discount these other things totally, but just not prioritize them, just because it it doesn't seem like there's a fit there, you know. And that's the best we can do with the limited amount of work that we have.
If we could just hold the questions, so finish the presentation. I've left plenty of time for questions at the end of the first presentation.
Okay. If something is not clear, feel free to ask me. But if you have a question, you know, if you could hold, that would be great. But if something's not clear to you in the slide, feel free to ask. So our team, you know, I've been talking a little bit about the work of the other teams. Our team has focused specifically on pesticides. And our question that we asked was, are chemical pesticides, particularly neonicotinoids, the most famous of which is imidacloprid. Some of you may have heard imidacloprid versus neonicotinoids. Imidacloprid is one of the neonicotinoids. Is this the factor, or is this the cause of or contributing factor to CCD? So that was the question that our team tried to answer. So our team consists of this very important person right here. This is Chris Mullen. He's a toxicologist, a chemist and a toxicologist who's worked a lot with pesticides in his entire career and has worked with neonicotinoids. Jim Frazier, this is my husband right here, who is an insect physiologist and has agreed to help us work on this problem, trying to figure out what these pesticides are actually doing to the bees. Sarah Ashcraft is a recent graduate and we hired her as a technician in our lab. Craig Sulla is our bee wrangler who helps us a lot out in the apiary, moving bees around, doing he and I together do a lot of the bee work. I know you're having some problems with bear fences, and although you can't see this very well, this is Craig Sulla's bear fence design, which is excellent. We have not had a bear problem since we've installed these fences. They're really wonderful, and if anyone wants to know about that, I can tell you a little bit about those later. So anyway, that's our team. And we work very closely with other teams, the most important one of which is this man right here, Roger Simons, who is the chief chemist at the National Science Laboratory, USDA National Science Laboratory. These people have unbelievable capability in detecting pesticides, detecting and quantifying pesticides. And they contacted us when we were trying to find somebody to be able to do this work. Roger himself is a beekeeper, which has been incredibly useful to us. He understands the language that we speak. You know, he has about 20 colonies of hives in North Carolina, and he makes a lot of sourwood honey. And so far, he has not agreed to share any of that sourwood honey with us, which is very unfortunate. I'm sure he will eventually. I'm sure we'll wrangle some out of him. And we work with the other members of the CCD working group. And we couldn't do what we do if they hadn't gone out and collected all these samples, you know, and sent them back to us. We also think that it's going to be very important to look at the combination of things like pesticides and virus. What we need to do is be able to reproduce on a predictable way, CCD. We need to be able to make it happen. Then we can say we know what the cause is. So we'll work with Diana Cox-Foster and the group working on pathogens to see if we can't do that. So the pesticide analysis, what we did was we looked at bee bread, we looked at trapped pollen. And this is the first thing we looked at. We sent those samples to the lab. Our chemist worked with Roger, Chris worked with Roger. They determined 171 pesticides that we would screen for, and they used the most likely pesticides that bees were going to come in contact with in agricultural systems, along with the beekeeper-used pesticides. We've looked at wax, brood nest wax of the CCD colonies. We've looked at foundation. We've also looked at brood, and we've looked at some other things like flowers, for instance. The source of our samples was the CCD study. So the samples that they collected out in the field from CCD operations as well as control operations. We also had a study where we took beehives from the university to three apple orchards actually. And those apple orchards, we knew the history of those apple orchards. We knew for the past 7 years what was being applied, and we knew what would be applied that season. And there are there's a study looking at reduced risk pesticides, trying to reduce pesticides and rely more on the natural enemies of the pests in the orchards. So there were two different blocks, two different kind of treatment groups in each of these three orchards, and those blocks were repeated 3 times. So we took colonies and we put them in those two different situations, the reduced risk where they were using these neonicotinoids, and then conventional blocks where they were using the pesticides they would typically use. And then we had a number of beekeepers give us samples from their colonies, either that were not doing well and they thought maybe they were having a problem, or that the colonies were in certain cropping situations, like corn, for instance. The bees were very near corn and were collecting corn pollen. Or they were in cucurbits, and we collected, or soybeans, and we had the beekeepers collect samples, or we collected samples using pollen traps. So Roger and his group, we prepare the samples, we collect them, we prepare them, we weigh them, we send them to Roger, and through some very sophisticated laboratory equipment, mass spec, GC, gas chromatography, HPLC, high pressure liquid chromatography, they shoot the sample in and they get these peaks out that can tell us exactly what the pesticide is and how much of it is there. And they can tell us at the parts per billion level. So very, very, very good detection of pesticides. Very capable. So what we found, and we are constantly sending samples, so we have additional data to add here, but this is the pollen that we looked at for 92 samples that we sent to Roger and his group. We found 43 different pesticides in that pollen and 5 metabolites. Metabolites are the breakdown products of the pesticides. Sometimes they're as toxic or more toxic than the pesticide itself. And you can see here the list. We have found pesticides in every class of pesticides. So the organophosphates, the pyrethroids, the carbamates, the neonicotinoids, the growth regulators. And some of these are not used at all anymore or have been phased out. So organochlorines and chlorinated cyclodienes, very, kind of hands-off type pesticides. Also herbicides, 13 fungicides and 6 herbicides. Some of these, a lot of these are systemic, which is one of the reasons why we were very interested in the neonicotinoids, because systemic means that the plant takes the pesticide up and the whole plant then becomes toxic to the insect that would feed on it. And we were worried about the pollen and the nectar, and were the bees collecting the pesticides in the pollen and nectar. So not only are the neonicotinoids systemic, but there are other pesticides that are systemic. So 14 of these were systemic. And we found as many as 17 different pesticides in one sample, and most of them had at least 5 pesticides in them. So what does that mean? It's bad enough to have one insecticide or pesticide, but what about the combination of these things? And we do know that certain fungicides, which I'll show you here in a minute, can synergize with certain groups of pesticides and become much more toxic. In one lab study, 1,000 times more toxic, the pesticide, the insecticide can become 1,000 times more toxic when it's present in combination with a particular class of fungicide. Only 3 samples of these 92 did not have a pesticide in them. So here's what we found in pollen, looking at the top, whatever that is, 20, 15 different pesticides in pollen, and what percentage of the samples had them. So you can see there, one of the most striking things is that these are the 3 highest, the ones that showed up the most often, coumaphos and fluvalinate were in over 50% of the samples. Fluvalinate in 72% of the samples, and coumaphos in about 58% of the samples. The other one that's highly used in agriculture that was present often was this clopyrifos. It's used in a lot of agricultural situations. Again, this is another way to look at the same information. This is the pesticide, the class, the LD50, in other words, how toxic these things are. This is how we measure toxicity by LD50. So the lower the number, the more toxic it is to the bees. This LOD is the level at which they can detect the pesticide. This is the range that we found, in other words, lowest to the highest level, the mean, and the frequency, how often we found it. So in the case of fluvalinate, this is maybe a bit of a surprise to you. That the LD50 for fluvalinate is 0.88. We're going to talk about that in a minute. That's quite toxic to bees. 0.88 means that 0.88 micrograms per bee, and that's what it takes. A very little amount to kill 50% of the bees in a 24-hour period. 24 or 48, I can't remember what the time period is. But we found that again in a lot of the samples of pollen. This is pollen. And we found it at from 2.4 parts per billion to 2,670 parts per billion, with a mean of 172 parts per billion. Again, chlorpyrifos, coumaphos were in quite high level in a lot of the samples. These are the two fungicides, this particular class of fungicide, that when present with certain groups of pesticides can make those pesticides very toxic to the bees. So that was a little bit of a concern that we found these fungicides in a lot of the samples. This is looking at the material by how toxic it is to the bees. So the same data, but how toxic it is. So bifenthrin is, you can see the LD50 there is 0.015, very small amount to be toxic to the bees. Thankfully, we only found that in 3 of the samples. This is why we're concerned about the neonicotinoids. You can see imidacloprid, 0.028, very toxic. And this pesticide, along with the other neonicotinoids, is used extremely widely. We're seeing these neonicotinoids replacing the organophosphates in everything from termite control to rose treatment, lawn and garden, backyard trees, orchards, golf courses, you name it. Animals are a lot of the flea and tick control now is actually imidacloprid. On the, I think, frontline, the active ingredient is frontline. So it's a systemic. It's in some ways, it really makes sense because you put the pesticide, a small amount, in the plant. It translocates throughout the plant. The plant becomes toxic to the insects that would feed on it. It's advantageous because you're not putting huge amounts. You're not spraying, big amounts of pesticide in the environment. You're being very directed. On the other hand, to things like pollinators, we are very concerned about them picking up this pesticide or coming in contact with it in the tissues of the plant, particularly in the nectar and pollen. Again, chlorpyrifos here concerns because we found it in so many samples, and it is fairly toxic. Fluvalinate and coumaphos, not as toxic, but still, because again we found it so frequently, it was a concern. In wax, now this is we're looking here just at the CCD colonies. We only looked at wax in the CCD and control operations. We didn't look at beekeeper collected wax or wax in the apple study. So we only have 18 samples here. But you can see in the 18 samples that we collected, 100% of the samples were positive for fluvalinate and coumaphos. Every single sample had coumaphos and fluvalinate. And you can see that the level, particularly of fluvalinate in the wax, in some cases was very high, with an average of 20,000 parts per billion, or 20 parts per million is what that would translate to. And we have additional samples, one as high as 200,000 parts per billion, or 200 parts per million. Oddly enough, that was one of the control colonies. So what we haven't seen, particularly in wax, is a correlation between sick colonies and high levels of pesticide, particularly beekeeper-used pesticide. And that's been a bit of a mystery to us.
So what's the definition of a control?
A control is an operation that didn't have CCD, and that where colonies were strong. So healthy-looking colonies, but there is one caveat, which I'll talk about in a minute with that picture.
But Maryann, what was the base on a sheet of foundation that was used? Have you tested sheets of foundation?
I'll talk about the foundation in a minute, okay? And let me just hold off on the foundation thing. Let me skip through this data with the CCD, and then we'll talk about the other things. So because we didn't see this correlation with high levels of pesticide, particularly in wax, and very, very weakly in pollen with strong versus sick colonies, we said, maybe we should need to look at the brood. Maybe we need to look at the bees themselves to see what the level of pesticide is. So we've only looked at 5 samples. We have a bunch of samples from the CCD study from control and CCD impacted operations that are at the lab being analyzed as we speak. But we sent these first 5 samples, and what we saw was that in the brood, 100% of the brood had levels of coumaphos and fluvalinate in them. And this level of fluvalinate here, and particularly at this level right here, the highest level, is near, very near, it's in the range of being lethal to the brood, given the LD50. So that was surprising to us, and that led us to think we need to look more at the brood. We need to look at the bees themselves to figure out, because all this is going on in the hives. There's pesticides in the pollen, there's pesticides in the wax, but it didn't really make sense with what was happening in terms of dying or weak versus strong colonies. So what we did then was we looked at, in the CCD study, we looked at we grouped together the dead, the weak, and the recovering hives against the strong hives to see if there was any relationship, any correlation. And what we have to consider, though, is that there was a lot of variability, in the pesticide levels, and that the problem with looking at the pollen is that if the colonies were CCD colonies, in other words, they were weak colonies with CCD symptoms, this happened, the pollen that we're looking at is probably not, unless it was left in there by the bees, it's probably not the pollen that the bees ate, because we're looking at, well after the fact, after the colonies kind of collapsed. That pollen was probably consumed a while back, and they probably collected different pollen since that time. So what we say is we're looking at these leftovers, and we're looking at this snapshot in time. We don't know what happened to those colonies afterwards. All we know is what they looked like at that moment. Maybe the strong colonies later collapsed. Maybe, who knows what the colonies that were doing a month before that. So this snapshot in time is a bit of a problem to not know what happened before or after. But what we did find was that when we grouped these weak, dead, and recovering hives against the strong hives, that we did see a higher level of fluvalinate in general. Again, it's highly variable. This shows you how variable it is. But the level of fluvalinate was higher in these versus the strong. And the same with coumaphos in the pollen. This is in the pollen. Is that right? Yes, in the pollen. So we did see this higher level of coumaphos and fluvalinate in those colonies versus the strong colonies.
Maryann, that's trapped pollen?
No, in this case it would be all bee bread, actually.
Okay.
It's all bee bread.
Okay.
It's all bee bread. In this case, we're looking just at the CCD study. And when we look at the brood, again, we're only looking at 5 samples. So it's, it's real hard to make a case here, but it's very easy to make a case for the fact that we need to look more at the brood and the bees. And you can see here that the fluvalinate, again, is higher in the weak, recovering, and dead hives versus the strong hives. In this case, also the coumaphos was actually, it was not as high. There was a slight difference here. It was lower in the and this is in the brood, of course, also. So we saw that in this case there was a higher level of coumaphos, slightly higher in the, although there's probably no real significant difference between this, but certainly here we're seeing a higher level of fluvalinate. So what is the issue with fluvalinate? I mean, we always thought that fluvalinate was a safe product for our bees.
Yeah.
Well, it turns out some things have happened with fluvalinate over the last 10, 12 years that nobody told the beekeepers about. That's very significant. And it's only because our chemist, who knows the EPA system and the databases and the literature, the chemistry literature, has gone through all this literature and combed through it and figured out what is going on with fluvalinate. It turns out when fluvalinate was first registered as a material for use, and they did the toxicity study for non-target insects, they found that it was, the LD50 was 65.85 about 67, there's 66 parts are micrograms per bee, that's a lot. You know, it takes a lot to kill a bee, in other words. It's a very low or high LD50, so it's non-toxic to bees, essentially. It takes a lot to kill a bee. But in the early 90s, there was a change in the way fluvalinate was manufactured. The chemistry of it is different. It went from something called racemic fluvalinate to tall fluvalinate. If you look on a package of fluvalinate, it'll say, or Apistan, it'll say the active ingredient for this product is tall fluvalinate. And what that did was it increased the toxicity of the material. It increased the toxicity of it to bees as well as to the target insect twofold. So in 1992, if you went to the EPA database and looked at the LD50 for bees, it's now 8.75 micrograms per bee. That's considered moderately toxic. And somehow, and I don't know what exactly happened from here to here, but if you look at the EPA database today and look at how toxic fluvalinate is to bees, it's actually identified as 0.2 micrograms per bee. This is from a number of studies that different people have done. And it varies, and that's why we use 0.88 in our, because what Chris did was he averaged several different research papers where they identified the LD, they did the work to understand what the LD50 was. He uses 0.88 micrograms per bee, but the EPA database identifies it as 0.2 micrograms per bee. This is highly toxic to bees. This means that we're using a material in our hives that is actually toxic to the bees, even in the strip. And furthermore, if you now, to make the material more effective, because fluvalinate's been around for a long time, and in the other ways that it's used, like for cattle dips and so forth, or even in some agricultural products, they mix it with something called PBO, piperonel butoxide. And it's to break down the defenses that the insects have to the pesticide to allow it to be more toxic. So if you were to use a fluvalinate product that had piperonel butoxide added to it, the LD50 now becomes 0.0094 micrograms per bee. Remember, the lower the number, the more toxic. This is extremely toxic. So fluvalinate plus piperonel butoxide is really a problem for the bee.
Is that an Apistan strips?
No, it's not.
Okay, that's off-label.
No, that's not.
Okay.
But, in other ways that fluvalinate might be used in a hive, if you used an off-label product, it's possible that piperonel butoxide might be there. Mm-hmm.
So the LD50 in 92 versus the LD50 in 2005, it was just it's the same chemical. It just looks like they were a little conservative.
Possibly, yeah. And I asked Chris about this, about why. I mean, this is more than twofold right here, and why. Certainly the big thing here was this change from the racemic to the tall fluvalinate. But the thing about fluvalinate, it's also being manufactured. I better not say this because I'm not absolutely sure. But there's some different people manufacturing it, some different companies. That doesn't, the registration has changed hands a number of times with fluvalinate, and so in the process of the manufacturing of it, each time they have to do certain, they have to establish the LD50, and somehow now, in the way that it's being manufactured, in the 1990s, it went from this relatively taking a lot to taking very little to taking very, very little today to be toxic. So, and I'm not, I think it has to do more with who's manufacturing it and how it's being used.
Is this PBO chemical in the environment that bees can bring it back and synergistically?
That's a good. It's a good question, and one that came up whenever we talked about this at the national meeting. And Roger Simons was at the meeting with the beekeepers, and we agreed we weren't looking for the piperonel butoxide, but we added that to our list of things to look for because we don't know. And it is possible that it's in certain other forms that is used in agriculture and might be in the hive in combination with fluvalinate. So what we have here, you know, in a nutshell, is the wax in our hive, from our work. We chemicals like fluvalinate and coumaphos because they're not getting those out in the field, and yet it's showing up in the pollen. And the fact that the agricultural chemicals are showing up in the wax means, you know, that we're not contaminating those agricultural chemicals other than probably by it coming in the pollen and somehow being exchanged with the wax. So there's this exchange of this fatty material. We've also looked at foundation. We have only looked at a very few samples of foundation, 5 samples of foundation from different manufacturers around the country, and or different bee supply houses, I should say. And we have found coumaphos and fluvalinate in significant levels in all the foundation that we've looked at. And again, it's only been 5 samples, and we have told all the supply houses, we've shared the data with them, given them the data, and told them that we would be happy to work with them to try to figure out what's going on here and can we reduce. Because certainly, you don't want to start with a pesticide. You know.
That throws off all your numbers right away.
Yeah. So that's an issue. Then we have the issue of brood food. You know, pollen is in the brood food, and if the pollen is contaminated, right off the bat, the bees are eating contaminated pollen. And also the question is the pesticides that are in the wax being absorbed into this brood food and possibly even into the eggs? You know, there are a lot of questions that we don't have the answer to. But in general, it looks like, at least from, and I have to say, you know, most of our work has been done with large commercial operations because of this CCD thing. And in general, it does seem that there is a pretty high level of toxicity in these hives. So in conclusion, we have this unprecedented amounts of fluvalinate and coumaphos at high frequency. We have numerous environmental insecticides coming in from fungicides and herbicides coming in from outside that are in both the pollen and the wax. There is a weak correlation or trend between CCD and in-hive miticides, although it is weak. And as you can see here, this has generated for us a whole lot more questions than answers at this point. So we have a lot of work in front of us, a lot to do. And we do have some recommendations for beekeepers, and I'll tell you a little bit about the studies that we have going on here. I don't want to go over my time, but we think that varroa mites are still a major player in trying to keep colonies healthy. And we think keeping varroa mites under control is a big... We've seen a lot of operations that have declined, and they have had high levels of mites. So this varroa mite thing is still very important. But what we are thinking is that the use of hard chemicals like fluvalinate and coumaphos is not, and amitraz, if people are using amitraz, although we haven't, I have to say, we haven't seen amitraz. The problem is amitraz breaks down very, very quickly. We are now looking, we have metabolites for amitraz, and we've started looking for amitraz in our samples. But we're really encouraging people to use the softer chemicals like formic acid, apagard, and apolite var, as opposed to coumaphos and fluvalinate. Monitor and control. We've seen a lot of people who have had problems use Fumidel B and feel like they've had that's really made a difference. They've seen healthier bee populations, less losses for the use of Fumidel. We are seeing this increase in Apis ceranae. We know that Fumidel is effective against Apis ceranae just like it is against Apis nosema, so that's to our advantage. We also think that replacing the pathogens and the pesticides are obviously building up in the wax. So this idea of culling old combs, be on a comb rotation, recycling combs, and or irradiation, fumigation of comb is really a good idea. Very good idea. So our work in front of us, our long list of things that we need to do is we need to figure out what do these levels of pesticides, either alone or in combination with each other or in combination with things like IAPV. What does it mean? How hazardous is it to the bees? Is it responsible for this decline? Is it responsible for this thing we call CCD? And how is it working? How is it actually, and how are the bees protecting themselves? In some cases, it looks like the bees are actually able to protect themselves from these pesticides. From our first look at this, we're worried about these synergizing fungicides, the ones that can synergize with pesticides and become more toxic, that particular combination of fungicides and pesticides. So far, the CCD work, the work to look at this big CCD study, we've each done our own little thing. And what we want to do, and what we are trying to do, although it's incredibly complex, is to look at the interaction between these different, what happens when you have these high levels of pesticide in combination with IAPV? Are those the colonies that are really collapsing? So this multifactorial, which Dennis Van Engelsdorf is working on. We're very concerned about the inerts, things that are added to the pesticides. We have now the chemical grade fluvalinate, in other words, 99.9% pure fluvalinate. Is it alone okay for the bees, but when we combine it with certain inerts, is that what's causing it to be more toxic to bees? And there is a lot of concern because the companies don't have to disclose these inerts. And we think in some cases the inerts might be the things that are added, the stickers, the soaps to make, the thing, or the things that make them more effective, like this piperonyl butoxide, are, rather than the pesticide itself, may be what's causing the problem. And again, we are trying to reconstitute, to reenact CCD in the lab and in the field. And we have some studies going on with gamma radiation to figure out whether or not the gamma radiation can be used to break down the pesticides. And so we think there is some hope that that could be the case. This is Sarah just in the lab showing you some of the setup that we have for doing these toxicity studies with the bees themselves, where she takes bees and treats them with pesticides and has controls and watches how long they live. That's sort of what it looks like in the lab. And we have a lot of collaborators on this project, long list of collaborators and a long list of funding sources, which has been great. Now we're going to add you folks to the funding as well for your contribution here. But the National Honey Board, the Florida State Beekeepers immediately came up with like $7,000 specifically for this work on pesticides. Tampa Bay, our College of Agriculture gave a big chunk of money to this work. A lot of beekeeping associations and individuals. We just got a grant from the USDA to continue this work. And there was another one I wanted to mention. Haagen-Dazs. Haagen-Dazs has given $150,000 to Penn State to work on, and particularly for public awareness. We're going to do, and some lab work as well, some research work as well. So that's been very, very helpful. And I got a call from a guy, they have a family foundation out in California, and they want to contribute $25,000 to this work. And they want to challenge their friends, their rich friends, I guess, to contribute to this work. I hope they're rich. I hope they're rich. So I'm going to stop there, and I'll be happy to answer any questions that you have. You had your hand up first.
You mentioned irradiation on one of your slides.
Right.
So we know irradiation eliminates spores, bacteria, fungus, other pathogens, but what effect does that have on pesticides?
Right. And we don't, our chemist, who knows how chemistry of pesticide works, thinks that gamma radiation should be able to, at some level, break down the bonds of the pesticides, making them less toxic. So we have a gamma radiation facility at Penn State. It's very small. They can only do small samples. But we've been taking samples down to them to see and then analyzing the samples before and after. And what we initially have found is that it seems that if we can, again, I don't understand the chemistry here, but emerging the wax in water does make a difference. If we can put the sample in water, that it seems to break down the pesticides. Very preliminary data. This is not going to work for you who wants to take, your hive bodies and put them through gamma radiation. But, for instance, for the foundation, it may be very possible to irradiate foundation. There's tons and tons of wax that's already contaminated, that if we can use this technique to clean up the wax that's used for foundation, it could be very, very helpful. So we're just getting started with that work. We got a grant, just got a grant from the Honey Board to do this radiation work to look at the degradation of pesticides.
Was there any thought, we know that bees forage in an 8,000 acre average area around their hives. Was there a thought to include as a control a hive known to not be within 8,000 acres of a person's lawn or a farm or something completely away from any of the
It's so tough. You know, the Forest Service is using these neonicotinoids in part to control, like for instance in the east, hemlock woolly Belgian. You know, it is very hard. I mean, we would love to find a controlled place, you know, where, but so far we haven't been able to do that. I mean, it's a good idea, yeah. Next. No, you go ahead and then I'll ask you. Yeah.
I'm originally from Ohio, and we have a very large population of Amish people. For those of you who aren't familiar with them, the Amish people, horse and buggies, no chemicals, no electricity, no machinery. And I thought that they were experiencing CCD just the same as we are.
Well, we have a large Amish population in Pennsylvania, and my understanding is that they do use pesticides quite a bit. That's my understanding. And, you know, they go to a lot of the meetings. They're getting credits for pesticide application. You know, we see them at these meetings. So I don't know that they're a good control. I think there are some, certainly. We have our organic meetings, too. You know, we see them there. I think they're, you know, some of them are using them, some of them aren't. So I'm not sure. I mean, I think our goal in part is to find some folks who are not using pesticides themselves in their hives, or bees are exposed, but it's very hard to really and truly find those kinds of operations. So far, and the organic operations that we know of, some have had losses, some have not. You know, that's our experience there.
I think there's a lot of research been done on all aspects of the pesticides and what have you. But one thing that we haven't looked into well enough, I think, is a comparison between the larger beekeepers that have these tremendous losses versus the small Northern queen breeder that has almost no losses. And I want to cite operations like our own, talking about just a few hundred colonies, not thousands, where the losses have been for many years between a low of 5% to a high of 10%. Now, that's such a tremendous difference. So when people ask me, well, you're doing so well, what are you doing? They're so different. And the only thing I can really say is that I haven't bought a southern package of bees in over 30 years. That's the only thing I'm doing different.
Are you using Kumafasi or fluvalinate?
We only use 4 chemicals. We use the Apistan, fumigillin, Paromycin, and menthol. Nothing else.
Interesting. Well, I think that's a very good point. We have been very focused on what's wrong, what's going wrong. And maybe the focus also equally needs to be on what about, as you described, what's going right. You know, what operations are, what's lacking or what's there in these operations that are doing well.
Especially if it's going on continuously. This isn't that for a few years we did well, then Bango was running.
Right, consistently. Yeah, I think that's a very good point. I think that's a very good point, yes.
A few things. One is, I think you've got some good job security.
This is not the security I'd like, though.
Secondly is, I brought this up at a meeting last year where scientists agreed to everything I said, but that's, but the third thing is, I think we have noticed the decline in bees ever since we've started using a tremendous amount of chemicals. And a lot of these chemicals were not used the way the labeling said they should be used. I'm using Maverick and fluvalinate and Apistan. All right, if we got away, and what I think the scientists have to do is they have to go back and try to breed that bee that is capable of surviving through the winters and not with all this pesticide stuff and all the chemicals we put in the hive. Natural hives didn't use chemicals. If the scientists started working like John Arbo, Jeff Harris, with their style of bees, or Purvis in Georgia, I think we would be better off and get rid of it. You know, and the testing of the chemicals before they're put on the market, they don't do all the testing that they should do. And I think we better get, the scientists better get there.
Yeah, I think you're right. I think resistance is the long-term answer to our problem.
I'm very sorry to see that fluvalvenate is one of the leading causes. I worked for the company that manufactured fluvalvenate as well as, in fact, it was in Gastonia next to the lab.
Oh, really? Is that right?
And also we manufactured the strips in our Dallas operation. And this was in the 80s, and we sold those operations out in the early 90s. So what you say about a possible formulation change could be valid, and it should be investigated, because fluvalvenate, the way I wasn't the original designer, but the way it was designed was a very good, safe product. Now the other thing to look at is what was mentioned here, is what we originally found out was the commercial guys didn't want to pay for the strips, but they did buy tanks of fluvalvenate and dipped rags and tunda process and stuff like that in it. So again, the suggestion was big guys versus home beekeepers. I think you'll see, or my suspicion is you're going to see some differences.
And to add to what you said, whenever these LD50s are established, fluvalvenate is used as a contact pesticide. As you well know, the bees and mites have to come in contact with it. So it's an external thing. When you dip a cardboard strip into a product, no matter what it is, it no longer is a contact, it's an oral dose. Right. They're eating, we see them, they're tearing those strips out and getting rid of them. So they're ingesting, which makes the pesticide much more toxic.
Our outfit that made the strips, originally what we made was flea collars. And that process was used to make the strips. So fluvalvenate, light doses, were compounded into the resin.
Right,
and that's how
it was obviously very effective. It's unfortunate that we had this resistance to it over time, which was inevitable with any pesticide. But, fluvalvenate, or Apistan, was a very good product, without a doubt. And there's just been this change in the situation. So, let me just...
Yeah, you were talking about the radiation of our hive bodies.
Right.
That over the years has included the frames, wax, pollen, anything left over, even some of the honey in the top of the frames. And it's been reinstituted after a hiatus now. It's using cobalt-60 at a fairly high level, about 15 kilograms. And over the years, hundreds of boxes of foul brood has been processed and put right back into operation. And some were done this year by George.
Yeah, it might be worth trying to look and see if there's lower levels in the material that's been irradiated. Pardon me?
It's done a facility that does a lot of medical stuff, so we bag it in the boxes.
Right. That's great that you have that.
Please repeat the question.
Okay. Yeah, he's talking about the gamma radiation and how you all have had access to that over the years for American foul brood control, and that it might be worth looking at some of that equipment that's been irradiated to see if there's lower levels of pesticides in that wax. Is that
yes,
what you're saying? Okay, great.
And also the pollen and the honey too.
Right. Although maybe after it's gone through, yes, that would be good.
I wanted to ask you about the possible contamination of honey with these pesticides and the hive miticides. I mean, it's not fat-based, so it's not going to hold as much. But did you look at some of that, and is there some of that?
We have tried very hard not to look at for obvious reasons. I mean, we have focused on honeybee health, and we have tried. We certainly were not interested in willing to look at honey.
We do think we need to look at nectar in the brood nest, and we have done a very little bit of that. And we haven't found very high levels, but we haven't done enough of it to really. But because of our work, one of the things that we did find was high levels of aldehyde, which is a serious human issue, in just a few samples in bees that were in orange groves being treated for a new disease, citrus reading. The levels were high enough in the pollen that if they were in human food, either honey or oranges, it could have represented a human toxicity issue. So we were duty bound to inform EPA and Bayer, who was also the manufacturer of aldehyde, about this level of aldehyde. They said that they initiated some effort to look at fruit and had not found aldehyde levels in the fruit high enough to be of a concern. EPA has now added honey to their list. Before they didn't analyze honey for pesticides. And because of this, it's now on the list, and they're now starting to pull honey samples from places around the country and look at honey for pesticides because of this work, for better or worse. I mean,
one other question. The Nosema cerani is still well controlled by the Fumigel?
Yes. Yes, Nosema cerani is controlled by Fumigel B. Yes, yes. Let me see somebody who hasn't had a chance. Go ahead.
Just from the slides, I think I misunderstood. I thought I saw a slide that said that there was no correlation between the high levels of pesticides and CCD. Is that right?
That there is a correlation between the sick and the wheat and the dead colonies and fluvalinate, did you say, or pesticides? I mean, specifically, the only one we found. Right. That's correct.
There is a correlation, or there is
It's a very weak correlation at this point. A weak trend, I would call it more of a trend of there being higher levels, particularly of fluvalinate, mostly coumaphos, pretty dicey, in those sick, dead, and recovering versus the strong colonies. Yes.
So is the conclusion is that it's alarming that it's there?
No,
That it's not causing
well, the conclusion is, our question is we need to do more samples. We need to see, is this real? Is this trend real? And we're doing that by doing more samples. We're also going to do that by following, there's been a migratory study this past year where Jeff Pettis from the USDA and Dennis Van Engelsdorf and their teams followed 3 migratory operations, 20 colonies in each operation, for a full year. Every time they moved those colonies, they went and they assessed the health of the colony and they took samples. And we're going to now take those samples and analyze them over time. And we'll see if the colonies that die, over time were exposed to pesticides, and we'll try to get a better sense of what's happened. It's real hard to make any hard and fast. I mean, basically what we're saying is the pesticides are there without a doubt. Beekeeper use pesticides particularly. What does it mean? And right now, it's really hard to say. Other than it's not good. We don't think it can't be good. So.
How strong is the correlation between the existence of CCD geographically? You mentioned that it's stronger in California.
Okay, so how strong is geographically CCD? This year, we're seeing, beekeepers have lost in the east bees too, although we haven't looked at our bees. Of course, a lot of beekeepers haven't looked at their bees yet. So we're just now the data is just rolling in. But it geographically doesn't really mean much because the guys in California are from all over the country.
I know, but what I was thinking was if it is a strong correlation, then you might want to take a look at the pesticides that are used in California. For instance, if they're using a lot of pyrethanoids on olives, for instance, PBO is a synergizer for all pyrethanoids. So getting back to Mary's good question, there could be a lot more PBO coming into the hives out from such a use.
Yeah.
And then of course those hives are going back over all the rest of the country.
That's right. That's part of the problem, of course, is that we move things so much. But yeah, I think that's one of the things we need to do is, and again with this migratory study, it's not so much geographic because it's been the East Coast. We've looked at the bees, the movement from north to south, and they're exposed each time they move to a different crop, and their likelihood of being exposed to different pesticides. We haven't looked, there's a lot of people out in the West who are doing work with, we just can't do it all, and so we need to collaborate with those people, we need to communicate with those people, but we haven't done an awful lot with what's going on in the West, but maybe we need to do more.
This is kind of a curiosity question. The people that you're working with out in the field, have they noticed any invasions from the Africanized bees into the CCD colonies?
No. And as a matter of fact, the Africanized bees don't seem to be impacted by this. Although, it's hard to say, because there aren't that many people who are keeping Africanized bees either, But no, they haven't seen any invasion of the dying colonies with African bees in Florida, for instance. No. Does that answer your question?
Yeah.
Okay.
On the track record of the country, how many of these chemicals are being brought in from foreign countries that we're not regulating and we don't know what's in there, selling them to whoever that are making the products?
Yeah, the question is about the importation of pesticides from other countries. And that is an interesting question, and it is a concern, because whenever some of these chemicals, we know they were manufactured, and maybe you might want to, the gentleman who was talking about, here, you left us, yeah, about the manufacturing of pesticides. More and more of that, as my understanding, is being done overseas as opposed to, and then there is concern about the technical grade of the purity of the chemical and the inerts. So yeah, there is a concern
The
about it.
additives that they're putting in this thing, we have no idea what they're doing.
The other issues, we're importing an awful lot of pollen from China. And so there's a concern about that issue as well. There's some movement to try to stop the importation of pollen. Although most of the importation of pollen is human grade, is human consumption, for human consumption that's used in bees. We actually have one sample of pollen from China that's at the lab right now to look at it for pesticides. But I think that's a legitimate concern.
When you're establishing the LD50 levels, do you have control groups of bees? Is it that chemicals are getting stronger, bees are getting weaker? How do you establish what that threshold is?
Yeah, the LD50s, most of the LD50s we've gotten have been from the literature on the pesticides that are, now we're trying to do some of our own work, and in that case, yes, there would be a control group, absolutely, and we'd compare the longevity of the control group against the longevity of the treated group. And in our case, what we'll do is we'll have treated with pure grade fluvalinate as you buy it with the inerts and controls, for instance. That's the kind of study we would do. And then you would apply it to the bees all the same, and you would look to see how long these different bees live.
And if you're saying that the so-called, whatever, I'm not sure how you're defining Africanized, but the so-called Africanized bees don't seem to have the same problem, has anyone done studies side by side of the pesticide effect on the Africanized bees versus the migratory?
Absolutely not. No, nobody's done really, very little work being done with Africanized bees other than what's being done, in the Tucson lab and the beekeepers out in the Southwest who are doing some work with small cell size and Africanized. So there's not that much work done. I'm sure there's been nothing done with comparing their
hardiness,
susceptibility to pesticides. Yeah, I'm sure that has not been done.
When are you going to publish your findings here?
We have a publication that we're hoping to submit to the American Bee Journal that we're just about completed, and then we are preparing a scientific publication, which hopefully, that takes a little longer, but the one in the Bee Journal, hopefully within the next 2 to 3 months at longest, maybe sooner, if we can get our act together.
And just quickly, were you testing trapped pollen for fluvalinate and cumulphos, or just the bee bread that was already in the hive?
Okay, we tested, whenever we looked at the bee bread and we looked at trapped pollen. For instance, we had pollen traps on colonies in the apple orchards and also in Dave Hackenberg's colonies in cucurbits because we were curious about that situation. And we had a number of other beekeepers who were in certain situations, particularly because we thought we would pick up the neonicotinoids. We had trapped pollen. And we did not find the neonicotinoids, except in the apple orchard colonies and the citrus greening situation. That was the only place we found neonicotinoids. And we did find coumaphos and fluvalinate in trapped pollen.
That's shocking.
So what does that mean? Well, if we found it in the brood, which we did, the bees themselves are likely contaminated with the product. So when they're out there collecting it and they're combing it off their bodies and putting the pollen into the, and we know they add enzymes and little bits of nectar to pack the pollen. Chances are they are contaminated and they are introducing the coumaphos and fluvalinate even at that point. So, yeah, I mean,
wow.
Yeah, not good.
Maryann, your report on the wax, that was in the American Bee Journal already.
I don't think so.
Yeah, well, I read it there.
No, we didn't put it there. I'm not sure. Maybe, when was that recently?
About 2 or 3 months ago. About the percentages in the wax and all that.
I'll have to go look and see. I don't remember that, but I'll look. Do we still, are we okay time-wise?
Yeah, well,
Okay.
Is anybody looking at the effects of or the correlation between larval hormone production, CCD?
Between larval pheromone production and CCD. You know, it's not new enough to look, I don't think anybody's looked at that particular aspect of it, you know, pheromone production in CCD versus non-CCD. I mean, it's so new, first of all, and, you know, we're talking a year. We've only had a year when we really recognized something as CCD, and the amount of work that has been done, given our industry, has been pretty amazing. There are lots of other things to look at, without a doubt, but it's just that this first year, you know, the work we tried to very quickly prioritize. No, I know. I see what you're pointing at. Right. So the pheromone production, is there something wrong with the brood? That's an interesting, and we have people who specialize in volatiles and could very easily do that, as a matter of fact, and who have agreed to help us with volatiles or which, you know, pheromones are going to be volatile compounds. They have very sophisticated equipment to do it. So that's a good idea. Yeah, I do have a website, and I'll give it to you at the second talk.
Anyone started studying actually the bats are dying off more than the bees?
Well, I don't know about more, but certainly there has been this big decline in.
Well, they said in some places there's 90% loss.
Yeah. And if you think about it, bats are insect eaters. So what's that? You know, is there a connection there? So
keep it down, Julianne. We're going to have a break in about 5 minutes. Keep it down so we can hear the questions, please. Stand when they ask the question. Okay. If you're posing a question, could you stand so the rest of us can benefit from it?
Yeah. I just want to make sure I understood one of your slides correctly, that you had a slide with all of the different pesticides, and did you say that there were a couple at the bottom there that were not even supposed to be used anymore? Or?
Well, it was, yeah, you know, I don't know my pesticides all that well, but there are definitely some pesticides that we have found specific pesticides that are no longer registered. No, it wasn't really, there weren't specific pesticides on that list, but there are some pesticides that haven't been used for a long period of time or haven't been registered for use that we picked up. So what does that mean? Does that mean that somebody used them and they shouldn't have, or are they in the environment?
Does that mean they're against the law?
What means that they're no longer registered, yeah. So effectively it would be that they were used, if there's no label registration and you use a pesticide, that's against the law. So, but it might be that they're also accumulated in the environment.
Yeah, so that's a mystery too.
Exactly right, it's a mystery. We don't know whether it was a misuse of a material or an accumulation in the environment. Another question?
Just to say, you know, I think the media did a great help to the beekeeper by last year putting all this stuff on television and the Discovery Channel, because you have people now that aren't even beekeepers that know you are coming up and asking you questions about CCD.
Right. I'll end on that note because I think that's a positive note to end on. this has really, I mean, you all have seen it in your increased attendance at your bee schools. People that, never, as soon as they find out you're a beekeeper in a grocery store line, they want to ask, what's up with this dying bees? What's going on with the bees? Have they figured out what's going on with the bees? So people are extremely aware. It has been, I have to say, though, a double-edged sword. I mean, in the balance of things, it has been very good for us, all this publicity. But we all have to do our part here, and we all have to help educate the public. One day, I was actually off of work. We had some friends who were visiting. I picked up the phone and I called my voicemail at work. I had 61 messages from one day. I sat and cried. How can I ever get back to all these people who are interested? And I wanted to, and the emails and the, people concerned, people thinking that they did something wrong, or how can they save the bee and hive in their yard. So in the end, I will say that this has been good for us, for our industry, but I think it has also challenged us all to become really, to make the effort to educate the public and share with people what's happening, what's going on, to be informed yourself, and to be prepared to go out and help educate people about the importance of a bee. This is our window of opportunity. The importance of bees, the crisis that we're in, the need to and maybe, there's a bigger message here about what we are doing to our world, to our environment. So I think we all need to be prepared to be spokespeople for the health of our bees, ourselves, and our environment.
We'll come back in 20 minutes.